Welcome to Ask the Expert. So digital cafe, very casual. And we're hosting Dr. Jessica Weaver. She's an assistant professor at biomedical engineering, the School of Biological and Health Systems Engineering at ASU, otherwise known as Arizona State University in Tempe. And her research centers on developing translatable cell-based therapies for the treatment of disease, with a focus on cell therapies to induce tolerance and transplantation for the treatment of type 1 diabetes. So the Weaver Lab uses biomaterials and immune engineering approaches with the aim to generate immunosuppression-free transplantation strategies. Dr. Weaver earned her PhD in BME at University of Miami with Cherie Stabler prior to completing a postdoctoral fellowship at Georgia Tech with Andres Garcia, and she was supported by the NIH Eyelid 2 training grant and the JDRF postdoctoral fellowship. And Dr. Weaver is currently a recipient of the NIH Director's New Innovator Award, which is really an interesting award and is a real shout out to what she's actually doing in this space. So I'm not sure why, but can you say hi, Jessica?
Yeah.
Okay, good. Now you took center stage there.
Okay.
So let's talk about what's going on in your lab. Now you have a long list of publications that came out of University of Miami with Cherie Stabler and also Camilla Ricordi. So some heavy hitters and some excellent papers. It looks like, you've got like kind of two things going on in your lab. You've got like Eyelid macro encapsulation design and immunomodulatory trophoblast-based therapy. You got two things sort of going on. Is that right?
Yeah, and actually, I prepared a PowerPoint. Should I share screen? Or?
Absolutely.
Okay. And I don't usually present from this office. Are you seeing the right screen?
Yeah, it's perfect.
Okay. I had a couple of screaming kids at home, so I had to run in here to give this presentation.
Wow, that's amazing. This is a beautiful image.
Oh yeah, this is when I was in Andreas's lab, we had the opportunity to do some really cool imaging. I have some more pictures in the presentation with showing revascularization of some of the islets that we transplanted. But yeah, I don't know, based on your email, I put together kind of like an overview of sort of the evolution of my research to what my lab's doing now.
That's great. And then the audience can chat about, what they're interested in with you.
Yeah, definitely. If anybody has any questions, just stop me. I tried to leave plenty of room, but we'll see. Sometimes I can be verbose. So, the main focus of my lab is really trying to eliminate immune suppression in cell therapies to treat type 1 diabetes, cell therapies in general, but the main focus has always been to treat type 1 diabetes. And so I'll be talking a little bit about that. Okay. All right, so my lab really focuses on using engineering approaches. We're an engineering laboratory and trying to use mainly biomaterials, but any sort of engineering approach to translate cell therapies as a treatment for type 1 diabetes. And so there's some critical challenges that engineering can be used to address that face the field of cell therapies. One of the big ones that I'm going to be talking about a lot is the need for systemic immunosuppression, which is probably, I would say, the biggest challenge. But there's been, some other issues that I've worked on over the years and continue to work on, including where to transplant these cells, so the transplant site. As we start to develop maybe devices to transplant these cells in, we start to have issues with nutrient transport. Islets are one of the most highly metabolic cells in the body, so nutrient transport, access to oxygen becomes a huge issue. And then there's a number of other issues related to trying to translate these into the clinic and different strategies to get these cell therapies into the clinic. And then of course, cell sourcing and donor shortages, which is of all of these challenges, the only area I don't work in, because that's a huge area unto itself. But of course, there's just within the last couple of weeks, we've seen a lot of data coming out from these cell therapies that are moving towards the market and going through clinical trials. So that's very exciting and definitely an area we'd love to collaborate with people in. Basically applying their cell therapy products in our devices and our strategies. But really what it all comes down to, and after about 14 years of working in this space, really the biggest hurdle to getting a cell therapy into the clinic is going to be this issue of chronic systemic immunosuppression. And really that's because the risk benefit doesn't make sense for the vast majority of type 1 diabetics, right? The side effects associated with systemic immunosuppression are much more dangerous to a type 1 diabetic than, especially if they're well controlled, the long-term side effects of type 1 diabetes, right?
And yeah,
and so that's
you know, they just had that big splash in the New York Times with the patient, you know, first in human, which is great, you know, but that was an older man, and a lot of, you know, newly diagnosed type 1 diabetics are children. So if you can't imagine like that level of immune suppression for a lifetime. So I totally, you know, this is to me, it seems to be the biggest issue.
Absolutely. And you know, compounding that, the autoimmunity makes it so much worse too. So we have to have special immunosuppression regimens just to get islets to engraft and function. And even then, I don't know if this number is out of date, but the last number I have for how long a typical islet graft, like a cadaveric islet donor graft, would last in a patient, or at least keep a patient off of insulin, the median graft life is only about 35 months. I think the data is a little bit better now with that, but I'm not sure what the new numbers are, but 35 months off of insulin. Now, they still have residual function, which is great, but it's that graft loss, that these underlying immune mechanisms and immune rejection that's going on that even with strong immunosuppression, we're not able to combat, right? And so really, if we could eliminate the need for systemic immunosuppression, potentially induce tolerance towards these grafts, then really it would be much more feasible to apply this as a treatment for type 1 diabetes. And so really that's kind of where all of my efforts have moved towards, because if we can't get over that hurdle, I still work in all of this other space, it all kind of goes together, but my biggest concern is getting past this immune barrier. And so just to kind of walk you through the last 14 years of how my lab has gotten to where it is now. And we're still a relatively new lab. My lab's only been open for about 3 years, about 2 of which have been during a pandemic. So it's been a bit challenging. But where I started was with Dr. Sherry Stabler at the Diabetes Research Institute in Miami, which is a renowned diabetes institute. So it was a really great place to start in the islet transplant space.
Yeah.
And a lot of resources, a lot of really people who knew a lot about the problem, right? And so the bulk of my dissertation, my graduate work in Sherry's lab focused on sort of the innate side of the immune response in islet transplantation, so battling inflammation. And so one of the main reasons that we focused on the innate immune response or inflammation in the islet microenvironment is because islets have a much lower innate ability to combat inflammation and stressors like that. They have lower levels of antioxidant enzymes, and so they're just not prepared to be thrust into a high inflammation environment. And so in traditional islet transplantation and how it's still done experimentally, I'm sure all of you know, is cadaveric islets are isolated from a donor pancreas and then infused into the portal vein of the liver, where they can spread out into this branching vasculature and become entrapped in this vasculature and hopefully secrete insulin in response to blood glucose. But the problem with that is we now know that there's this instant blood-mediated inflammatory response. So this instant inflammatory response to the islets, and the islets are not equipped to handle that inflammation. And then on top of that, you have all of these mechanical stressors that islets are also not prepared for. They don't see that normally in the pancreas. They're shoved into a little vessel, and then they're constantly getting pounded by the blood, by blood pressure. And so all of these things combined lead to, now we know about 60% of cells lost almost immediately upon transplantation. And so essentially when we're infusing islets into the liver, we're practically throwing them away, right? This is not ideal. And that's why it takes between 2 and 4 donor pancreata typically to reverse a type 1 diabetic and get them off of insulin. And so that's not ideal. And so Sherry's lab, I was one of the first PhD students in her lab. she was really focusing on, where can we put these islets and how can we deliver these islets in a way that's outside of the liver and in the context of less inflammation, and hopefully preserve more of these islets that we're transplanting. Ideally, 100% would survive the transplant process. And so some of our first work in this space was, Sherry's lab started with developing these silicone porous scaffolds that the islets could be seated in and then transplanted into a non-liver space, so tissue outside of the liver, extrahepatic tissue. And so we used a mouse model, and we used the epidermal fat pad, which is essentially the murine equivalent of the human omentum, which is a highly vascularized organ that isn't usually talked about in anatomy and physiology courses. It's considered vestigial because they don't know what it does, kind of like the appendix. But it's got some interesting features that it may be a little bit immune privileged. It's very highly vascularized, so it has some features that might be positive for islet transplantation. And so we delivered these islets and these scaffolds to these epidermal fat pads and saw reversal. And then to take it a step further to combat any possible inflammation that might be seen in the site, perhaps in response to the scaffold that they're delivered on, or just the trauma of transplantation itself. We incorporated a really potent anti-inflammatory into this transplant site to locally deliver low doses of an anti-inflammatory and prevent this inflammatory response. And it was pretty successful. We saw earlier reversal of these syngeneic transplants, and Sherry's been taking this a step further with different variations on this delivery method that have proven to be pretty successful. And then the other half of my dissertation work, again, still focused on this problem of inflammation, was developing this really interesting encapsulation material. And so just to kind of some brief background on encapsulation, I'll be talking about more a little bit later. But essentially, encapsulating islets has been around as an idea for about 50 years now. And the premise is that you would put an islet within a material, so typically a hydrogel, that would cover the surface of the islet to prevent direct contact between that donor tissue and the host immune system. And if you can eliminate that direct contact, then the idea initially was that you could eliminate the need for immunosuppression. We now know it's a little bit more complicated than that. Encapsulation by itself is probably not going to do the whole job, but it still does a really great job of eliminating that direct antigen presentation. So it does something. And at the time during my dissertation, with this focus on inflammation, Sherry had stumbled across this really interesting nanoparticle system called cerium oxide nanoparticles. That it's basically a crystalline nanoparticle that acts just like antioxidant enzymes, superoxide dismutase and catalase, and that it's able to uptake these free radicals like superoxide and convert them into neutral molecules like oxygen, and in the case of catalase, take hydrogen peroxide and convert it to oxygen and water. And so the idea was that if we could create this nanocomposite hydrogel to encapsulate the cells in, we could create an antioxidant barrier that would prevent any of these byproducts of inflammation, these free radicals, from reaching the cells and preserve their viability. And it was actually,
Jessica, where were these allogeneic capsules? Where were they? What context did they come from? Were they in another disease treatment, paradigm, or was it just like they were,
No, I think the first was in the context of islet. I think as far as cell transplantation I might be wrong. I think islet transplantation was really just the first step towards delivering just a cell and trying to protect it. It's amazing how early the idea of encapsulation came about, and it's amazing that over 50 years it hasn't changed all that much.
Yeah, but how about the nanoparticle piece, the crystalline nanoparticle? Like that is really, that's totally advantageous, right? You get rid of all those ROS and just release oxygen. But
and it was very successful at scavenging. You can see in the image at the bottom right, we hit them with a pretty high dose of superoxide, basically destroying our controls. And it was able to do a really great job of scavenging. And what's actually the best feature of these nanoparticles is that because of how the crystalline structure of these nanoparticles works, it's actually self-renewing. And so it constantly switches its valence of the different seria within the structure of this crystal, so that theoretically it has an infinite half-life. And so it would never run out of,
yeah, that's amazing. And is that are those nanoparticles used in other contexts outside of diabetes? Is that where they originally were being used, or is this like brought in?
I'm not sure where she ran across this system initially, but yeah, it's used now in a lot of industrial contexts. It's just a very useful system in a lot of contexts. Yeah, and I've seen quite a bit. It's actually surprised me how much it gets cited more and more every year. So I think people are finding more and more utility in tissue engineering for these nanoparticles as well.
Cool.
Yeah. And so that was sort of the two arms of my graduate research with Sherry Stabler. And then when I wrapped up that, and she's continued both of these projects, continuing to optimize and refine and come up with better ways. Sherry does a lot of like nano encapsulation, so sort of layer by layer surface stuff. And I believe she's relatively recently published sort of the next iteration of this as well. So she continues to work in this space, and we've always talked about potentially collaborating on the serious stuff again. I just haven't had the bandwidth to go back to using it, but it's definitely a very promising system. And so after that, I had the opportunity to go to Andres Garcia's lab for my postdoc at Georgia Tech. And so Andres, his lab is primarily focused in biomaterials, developing synthetic hydrogel systems in the context of tissue engineering. And he was really interested and had just recently, by the time I joined his lab, gotten into applying these hydrogels in the context of eyelid transplantation. And he brought me on because he wanted someone who had a good amount of knowledge base in the eyelid area, just because there's a lot of niche, assays and things like that, transplants, isolations, things like that. So this was a really great opportunity to come in and work with a new hydrogel platform, as well as have the freedom to investigate some questions that I had in the eyelid transplant space. And this first paper that we published was really kind of answering a question I had, which was, in Sherry's lab, we started using these extrahepatic transplant sites, this epididymal fat pad. But I was seeing at the same time, a lot of people were developing platforms to deliver, for devices to deliver islets under the skin, like ViaCyte's device.
Right.
But a lot of people were looking at the subcutaneous space because it's minimally invasive. Other people were
and you can remove it. And you can remove it if there's like a teratoma or some kind of problem, right?
Mm-hmm, mm-hmm. And so my question really was, well, we know that you lose like 50 to 60% in the liver. The liver is a hostile transplant site, right? So do we know though, or my question was, are one of these extrahepatic sites going to be superior to the other? And is there a way that we could directly evaluate, is one of these sites better and why, right? Mm-hmm. And so this hydrogel system that came out of Andreas's lab, using these synthetic PEG hydrogels that we could make degradable, non-degradable. It's a very versatile synthetic system. We can incorporate signaling into it, like vasculogenic signals to encourage vascularization. And we can, it cross-links in situ. So we can use it to deliver cells and deliver them in a degradable hydrogel that would hopefully remodel and ideally the islets would revascularize. And so we were able to use this to directly compare in parallel different transplant sites to see if there were any differences in how islets engrafted, anything like that. And this was really answered a lot of questions for me. And what we found was that the vasculogenic hydrogel worked great. The islets did vascularize even without the VEGF, but the addition of VEGF definitely accelerated the revascularization process. We got these really beautiful, like large clusters of islets that all revascularized pretty quickly. In these pictures, you can see some GFP islets and some non-GFP islets that have been revascularized in the EFP. And when we directly compared the sites, so particularly sub-Q versus the omentum or epididymal fat pad in the mouse, what we found was that there was a higher degree of innate or inflammatory immune cells in the skin site versus the EFP, which in retrospect makes a lot of sense because it's a barrier organ, right? So it's got to have good surveillance to prevent, microbes from penetrating the skin. And so that made sense in retrospect. And additionally, what was a little bit surprising was that the subcutaneous site had a lower starting point with the degree of vascularization in that site relative to the epidermal fat pad. And really, it was sort of that baseline amount of vascularization that we started with that impacted how much vasculature we were able to get and how vascularized we could get these eyelids. Mm-hmm. And so what was interesting was with the vasculogenic hydrogel, we did see improvement over the hydrogel without any VEGF, but not enough to get a single pancreatic donor functioning in the recipients, which was our big goal, right? It's in the past with the liver transplant, always takes like multiple pancreata. The real goal here is preserve as many islets as possible in the transplant procedure so that we can get a single donor functioning in a patient and get that patient off of insulin, which we really weren't able to do in this context with the subcutaneous space, but was relatively easy in this EFP, especially with vasculogenesis. And so that work really answered a lot of my questions. And from there on out, I've been very focused on using the fat pad. People have definitely shown it's possible to get mice off of insulin or cure mice using the sub-Q space. I think the barrier is just much higher to do it in the sub-Q space. And that probably the focus needs to be on using a site that's more conducive to islet survival and function. And so at the same time, Andres's lab was using these synthetic hydrogels to develop non-degradable microcapsules, very similar to the Allogeneic microcapsules that have been used for about 50 years. And a talented grad student in the lab, Devin Hedden, had developed this microfluidic system to generate these small PEG-based capsules that were a bit smaller than the traditional Allogeneic capsules. So they were about 500 microns in diameter. And the advantage of that is that we reduce the transport distances of oxygen getting to the cells, and we have much better, hopefully much better survival of encapsulated cells. But the real big advantage to me by reducing the volume of material that you have when you have a graft is now the graft size is reduced substantially. And so now we could start thinking about where we could put these encapsulated cells, not just throwing them into the IP space, which has a lot of safety issues and is sort of the traditional way that people transplant these cells. And so we wanted to try and use this vasculogenic hydrogel in conjunction with these capsules in a defined transplant site so that now an encapsulated graft is retrievable. And so we delivered them with that vasculogenic hydrogel and got really, really nice vascularization on the surface of these capsules. And the idea is hopefully that if you can establish a really high degree of vascularization at the capsule surface, then you can maintain that level of oxygen at the surface of the capsule and have this continuous oxygen supply. And compare that to the IP space, which is the traditional space that people put these capsules, where eventually, no matter the material, you get this fibrotic buildup on the capsule that's just kind of floating in the IP space, and you start to wall off from localized oxygen. So the goal here was really to see if we could harness the remodeling of the tissue and optimize how much oxygen we could get to these encapsulated cells. And this worked very well. We got comparable function to Allogeneic capsules and comparable function of PEG EFP compared to Allogeneic IP. And sort of at the same time, while the reduction of material is great and that's been a goal to improve transport, you still have all these individual capsules, which is a concern because even though we're trying to squeeze them all into this defined transplant site, inevitably we saw escape of some of these capsules into the IP space. So we're still facing a safety issue, right? Yeah. And so simultaneously, we were working on this idea of macro encapsulation using these PEG hydrogels. So the idea being we could contain all of these cells or many cells in a single device that would then be easily retrievable and not worry about losing islets into the ether. And
Have
so
you guys
orthodox-
ever, did you guys ever consider a collaboration with Amir Dolan at University of Galway in Ireland? She's got that dynamic soft reservoir that oscillates, and she's got this whole thing where, you know, it's supposed to really, the oscillation itself, it creates enough fluid flow to alter that environment around the implant and protects against fibrosis. It's really kind of cool. And you know, she came out of MIT, I believe. She was you know, given she got the like 30 under 30 or whatever they call it at MIT. But it's a really interesting, it seems like it, you know, you guys would be a natural collaboration. Yeah.
I'll have to take a look. I off the top of my head, I don't recall seeing that, but I may just not remember. So I'll have to go take a look at that.
Okay, sure.
And see how that system works. I do know some investigators with sort of similar, trying to design devices where you have continuous flow to keep, you know, transport at an optimal level for larger devices like this. It's definitely a lot of work in this space.
Yeah.
Minglin Ma is working in very similar sort of mindset as my lab.
Yeah, talk to him.
I'm always excited to see his work come out because it seems like we're doing very similar ideas, which means I think we're on the right track.
Yeah, he came out of Dan Anderson's lab when they were pursuing Sedulon.
Mm-hmm. And so
he's got that background.
This macro encapsulation strategy, we had sort of the same idea that we would potentially try and vascularize the surface to improve transport, improve viability. And while we did manage to get, you know, similar to over here, really nice vascularization at the surface, and in this case, it was actually in a rat omentum, so the true omentum, in a larger animal model. Then we did see improvements with more vascularization in viability of the cells within the device. We did not see good function out of these devices. Part of that was probably just transport of insulin out of these hydrogel devices, but ultimately, really, it came down to oxygen. You know, one of the big features is always going to be oxygen with these cells. And so this is a big focus of my lab now. You know, I really believe in macro encapsulation being the forward direction of this, trying to get cell therapies into the clinic, just from a safety perspective and even from a manufacturing perspective. So how can we get cell therapies, including these stem cell derived products, you know, into patients, and just thinking a lot about how this would work in the clinic and how it would work in a cell manufacturing context too. And so my lab does a lot of thinking about oxygen transport, modeling oxygen transport, and thinking about how we could improve macro encapsulation design so that we get better transport to the cells in a comparable way to microcapsules, right? And so we think a lot about how we can generate high surface area to volume ratio geometries that would have similar transport to capsules. And so one example of that is like this spiral design here. And so as we started to think about these complex geometries, we also started to think about, well, how are we going to make these, these complex geometries? And so we developed this hydrogel injection molding system that's basically like a handheld system to generate these complex hydrogel geometries.
I love the
and it's a relative
ones.
What?
I love the ASU ones. Those are great with little tray.
Yeah.
Perfect.
You always have to rep the university, right? And
that's right.
We've got our little
absolutely
Sparky trident here.
Fantastic. Yeah, this is really interesting now. What you're talking about here.
Yeah, and so the idea behind this was to, you know, we have kind of two upcoming applications. One being if we want to do actual primary human islets, we would have to have a means of delivering these encapsulated islets or generating these macro encapsulation devices at the patient bedside. So the doctor receives the islet prep, and then he can encapsulate it at the patient bedside. And we wanted something that was easy for a clinician to use, sort of intuitive, and this injection molding process really is that. And so just about anyone can generate a complex geometry using this injection molding. And then even further than that, thinking down the line with cell therapy, so these stem cell derived cell products, is how could we have a high throughput sort of manufacturing of these encapsulated cell devices. And so injection molding is very amenable to high throughput manufacturing and automated manufacturing of typically plastic objects, right? But in this case, we can use it to as basically a biofabrication technique, and hopefully we think that this would scale really well with these new products that hopefully will be coming out soon.
How many cells can you fit into Model D?
In model
0.5 millimeter.
Oh, what, like
the one that looks
the
like
blood vessel?
Yeah, like a blood vessel.
Well, that's really just more for fun, the blood vessel thing, just to show the different like physiological. I don't know how practical the vasculature. So we haven't played with loading cells into this. For the eyelid application, we're really focused on sort of this spiral geometry, just because it maintains that high surface area to volume ratio of capsules, while also having just a compact geometry, and also having these gaps in between the arms should allow vasculature to then wrap completely around each of these spiral arms. That's the hope.
Can you get into the 2 millimeter piece? Could you get an entire set of eyelids from an adult human pancreas, or no? You need
our initial designs here are for a modular device. So starting to think about just primary human eyelid preps. You never know how many eyelids you're going to get from a human pancreas, right? And so we've designed these to be modular, where we're trying to get as many as we can into a single, but shooting for around 100,000 IEQ per device. And that way, if you get a pancreas and let's say you get a fantastic yield of 700,000 IEQ, then you can pull 7 of these kits off the shelf, and you can do 7 individual spirals, and then the dimensions should be small enough that you can fit them all into a single omentum. Or if you only had 300,000 IEQ, then you could grab 3 and make 3 spirals, for example.
That's great. Okay, thanks for going through that.
Yeah, so that's the general idea of this approach, and we've tested it pretty extensively on single cells to make sure that this process isn't killing the cells. Really, the forces that the cells are seeing are very similar to other biofabrication techniques like 3D bioprinting, things like that. So we weren't expecting too much cell death, but we wanted to double check, and especially because islets are, incredibly sensitive cells, right? And so we even tested this on primary human islets, and we found that the injection molding process didn't appear to damage their viability or their function. And so we're very pleased with that, obviously. And so now we're moving this into our preclinical models. The COVID slowed us down a little bit, but we're finally getting into animals.
Fantastic.
And so doing some, I'm sorry.
No, I said fantastic. I think there's a question in the audience. Is the spacing distance between each spiral an important parameter in the oxygen availability profile?
It has not been in the models that we've run, but it's really going to depend. There's only so much you can model, right? So we need to see what happens when we put these in vivo and see whether there's an optimal just for how the tissue remodels, and whether perhaps the vessels prefer a certain gap to actually get a certain size vessel through there. So we haven't really played with that extensively yet. We're still in kind of early stages and investigating how these vascularize in vivo just now and how they function. We have some promising data so far in vivo, so we're happy with that. We weren't expecting to have too many troubles with it, especially because what's great about this platform is it's pretty versatile. We've managed to get it working with different hydrogel platforms, so Allogeneic, PEG, and so using traditional Allogeneic, we didn't really expect to see any issues with function, and so far it seems like primary islets function comparably to individual microcapsules of Allogeneic. But we have to do a lot more studies. This is very preliminary data.
That's still encouraging.
Yeah. Yes, we're happy about it. And so kind of continuing in this area, alongside of our preclinical studies, we also recently started well, not really recently, we've been working on this for a little bit, but we have a collaboration with Dr. Vikram Kodabagkar, who's also faculty here at ASU, And the Cotabacar lab develops these oximetry probes, these magnetic resonance oximetry probes. So basically, these probes are able to give a readout of the oxygen tension in vivo longitudinally. And so we are really excited about this because obviously we care a lot about oxygen.
Yes.
And so we've been basically developing these hydrogel composites with these oximetry probes to investigate the actual longitudinally the oxygen within these devices after we transplant. And so we're just about to put this into animals as well. And we've gotten everything kind of optimized in vitro, and we're really excited to see whether we can in real time delineate the differences in oxygen between different kind of encapsulation devices and even different transplant sites. And so kind of coming back to my postdoc work, half of my lab is very focused on encapsulation as a means to hopefully limit how much immunosuppression we need. We know now, though, that encapsulation on its own is not going to eliminate the immune response. That was the original idea of encapsulation, but we've learned a lot more in immunology since the idea originally came up. And so just for a little bit of a background, how tissue is normally recognized when it's transplanted, if it's not encapsulated, is through two different pathways, but one of the strongest pathways is through direct contact, right? So host immune cells are directly in contact with the surface of the donor islet, or perhaps a passenger APC, antigen presenting cell that was on the islet, and that initiates the immune response. But even when we encapsulate islets, no matter how we design these encapsulation materials, if we want to design them for rapid efflux of insulin, inevitably we're going to have rapid transport of antigens out of this capsule, because there are many antigens that are smaller than insulin. So there's really no way that we're going to be able to design these capsules to prevent antigen shedding, which will inevitably be picked up by host antigen presenting cells and initiate a immune reaction.
Yeah,
it's definitely
they have a memory. Yeah, they have a memory of
you know. It's not even just the autoimmunity, although that is awful. Yeah, a huge barrier in islet transplantation. But just initiating this, and so it's harder for them to home to the cells because it's more difficult for them to find them. But some of my data, particularly that American Journal of Transplantation paper with the microencapsulation in the EFP, we did a really interesting study where we transplanted transgenic luciferase expressing islets and tracked their survival, their actual viability long term. And we found this sort of gradual destruction of the islets, which was sort of like, to me, sort of direct evidence of this sort of smoldering immune response that you get against the islets, even with that capsule in place. And so that's been kind of known. And so there's a lot of work in trying to directly modulate the immune system, whether in conjunction with encapsulation or just as a strategy on its own. And so some of the other work that I had the opportunity to be a part of in Andres's lab at Georgia Tech, and in collaboration with Haval Shirwan and Esma Yolku, who I still collaborate with now, who are immunologists. So the Shirwan and Yolku lab had developed this fast ligand engineered protein that enabled us in this collaboration to now tether these immunomodulators to biomaterials. And so the two main immunomodulators that we investigated was fast ligand and PD-L1. And these are two molecules that are very common in cancer. And so the original idea was basically to mimic how is cancer hiding from the immune system, right? And a lot of people go to cancer as a model because it has immune escape, right? Yeah. And so we saw some pretty promising results just using biomaterials presenting these immunomodulatory proteins within the transplant site next to the islet. So not encapsulating the islets. These islets are fully exposed. but having basically an off-the-shelf immunomodulatory material that we could deliver with the cells to prevent the rejection. And we did this in combination with a short course, low dose immune suppression based on some of the Shirwan Lab's past work investigating this molecule. And with that combination, we were able to get around 50 to 60%, depending on fast ligand or PDL1, long-term survival, relatively long-term survival of these grafts. And so that was very, very promising. We're very excited about that. And some of the, I haven't seen some of the further data that's coming out. I'm looking forward to the paper because it's been since I left, but I think this work has continued to show promise. But as I was starting to think about how I wanted to approach generating tolerance against the islet grafts in my own lab, I started to think about, this model system that people typically use, which is cancer, right? And while cancer does a good job of immune escape, it has kind of a low bar to escape from the immune system because it's your own tissue, right? And so you don't have this MHC mismatch like you have in transplantation, and definitely don't have the autoimmunity, right?
Right.
And so it's not as high of a bar for it to hide. And so while these molecules definitely showed some promise, you can see that we're not completely escaping this immune response. And we still needed a little bit of immune suppression, which is not ideal. And so that led me to think about, what model systems could we use that would be more, would be a stronger model system. And so that got me thinking about basically the only system wherein you have in human biology, where you have a completely allogeneic tissue not rejected by the host. And that the only system where you have that is pregnancy. Now you have some systems in our biology where semi-allogeneic cells escape immune rejection, right? So testes and ovaries, but those are not fully allogeneic and they're single cells, right? And
right.
We're thinking about like a full organism escaping immune attack. And an allogeneic one as well in the case of surrogacy, fully allogeneic. The only model really is pregnancy. So it's a really powerful model.
I
and
agree
so that
with
got me
this completely.
And so and it was surprising to me that there wasn't work in this, not a lot of work in this area, or trying to translate these mechanisms. A lot of it was focused on cancer. And so that's really been the focus of my lab is basically we call it placental mimicry. And so the premises and how the placenta works, and the placenta is really the mediator of this immune tolerance against the fetus. And so the way the placenta works is it consists of, it's completely fetal tissue, and it consists of these cells called trophoblasts. And so these trophoblasts are primarily what is in contact with the maternal tissue. And these trophoblasts, really, really interesting, I found, very similar system to our encapsulation system, wherein these trophoblasts essentially present a inert surface to the maternal tissue. And so they don't present classical MHC, HLA, ABC.
But they have HLA-G.
They present HLA-G, HLA-E, which have these sort of tolerogenic, they're not as well understood as the classical HLA, I think.
Right.
I still keep doing deep dives trying to understand how HLA-G works. But basically, they certainly don't activate immune cells against these tissues, and even promote a tolerogenic response to these to this placenta. And what's really interesting, though, is it's similar to encapsulation. We're presenting this inert surface. The placenta cannot prevent antigens from being shed to the maternal side. And so, again, just like encapsulation. And so what's interesting is how the placenta modulates this is that these trophoblasts secrete a suite of tolerogenic factors that educate the local antigen or the local immune cells, so antigen presenting cells that are scavenging these antigens, educate them towards a more tolerogenic phenotype, so that what results is not a hostile antigen recognition of this tissue, but actually a tolerogenic, antigen-specific tolerogenic response towards this fetal tissue, which is exactly what we are looking for in, delivering encapsulated cells or even unencapsulated cells. And so a big focus, and at least 50% of my lab is focused on ways that we can use this model system and some of the mechanisms that the placenta uses to avoid escape or avoid rejection from the maternal immune system. And so kind of in the initial steps towards this in this direction, we've actually got a great collaborator in trophoblast biology, Mana Paris. And so she's definitely going to be a lot of help as we dive into this new space.
Where is she located?
She's at UCSD.
Okay.
Yeah, and she's worked for years in this area, and so she's been very helpful already. And so as kind of an initial step, is what we've been doing is taking these trophoblasts and trying to generate sort of complex organoid. So basically little artificial placentas in a 3D biomaterial tissue culture platform. So trying to generate structures that are similar to what we would see in the placenta, and play with the architecture of these organoids that we're generating in the lab to see what kind of immunomodulatory mechanisms these cells have. We're really interested in teasing out the different immunomodulatory factors that the different cell subtypes secrete, because there's at least 3 subtypes of trophoblasts, and it's unclear to us which one would be optimal. If one of them is more immunomodulatory than the other, what different factors do they secrete?
Could you re-engineer these types of cells to be insulin producers? If so, which ones? Ekaterine Bershvili over there in Geneva is, her team, her group is looking at insulin producing organoids that they engineer from eyelid and amniotic epithelial cells. Her postdoc is Fanny LeBreton. She's on the cutting edge of that. But yeah, you guys have a lot in common in like what you're thinking about, which I love. And hopefully, you'll both be there in our upcoming State of the Science, which is featuring women scientists. I'd love to hear you guys talk about, your approaches together, because this is so, I just think this is such a fascinating model system.
Yeah, we're very excited about it. And this, as you mentioned before, this was what I got the new Innovator for, kind of a unique approach to trying to basically eliminate immunosuppression. Initial stages, the initial plan is to integrate this with either encapsulated islets or ideally unencapsulated islets, because a lot of my past work has shown how critical it is to vascularize islets. And then ultimately, we have somewhat of a plan to generate essentially a tolerogenic vaccine using this system, where we could potentially induce this tolerance prior to the delivery of a graft. Now, this would work fantastic with a stem cell derived cell therapy for type 1 diabetes, right? Because we would know the haplotype of the tissue that we're transplanting before we do it. And it would work really well with live organ donors like kidney, things like that. Probably more challenging with any cadaveric donors, but we'll have to see where this goes. And it's, again, very pie in the sky. That's where we'd love to get to. And we hypothesized, and the reviewers thought that it was possible. So yeah, we're trying it.
I mean, it's very innovative. I mean, you've got some, proof of concept here that this could be a really exciting space. And I cannot wait to see where you go with this. I mean, it's a really neat system.
Yeah, we're definitely very excited. I'm glad other people are excited too. And I hopefully any trainees, postdocs, grad students out there who are looking for a position working on some interesting stuff, we have opportunities available both on that project, so the placental mimicry, as well as encapsulated islet oxygen in vivo. We have a collaborative R01 on that MRI oximetry project. And so here's just recently we did a little gingerbread house making contest. So some of the students made it out to that. And, obviously wouldn't be here without my fantastic students that are generating a lot of this data. So
it's a fun outside the lab engineering project. Cute.
Yeah, exactly.
there's a question from someone in the audience, and she's wondering about, so it's feasible that females with type 1 titer out high antibodies to their own beta cells may not attack fetal insulin. Does fetal insulin work as well? Huh. I don't know. I don't know that answer, obviously. If anyone in the audience, anyone else knows, it'd be great, but
I don't know.
I'm curious.
It's interesting. I don't know much about that.
Yeah, I mean, it isn't totally in this wheelhouse, but it could, I mean, it might have some impact on that whole fetal maternal interface. Who knows? Well, maybe the next time we speak, you'll have stumbled across something that's of interest there. But this is an amazing project, amazing approach. And I wish you guys, speed and discovery, and look forward to speaking with you again soon.
Thank you so much.